I won't do that. I won't do that. Hello. Please take your seat. Welcome back. No, it's a real privilege to introduce Amy Zanne, who is a RSC chair in tropical ecology at University of Miami. Amy explores how plants, microbes, and insects interact and how this impacts the carbon cycle. She's worked in Australia, South America, Africa, all across the world on tropical ecosystems. So she has a broad experience on many topics. But she'll be talking today about termite sensitivity to temperature and how it affects global wood decay rates. So Amy, welcome, and the floor is yours.
Thank you so much, Jerome. And thank you so much for everybody for being here and inviting me. I tried desperately last year to join you, and this year is the one that works. So it's a treat to be here with all of you today. Let's see if this is. So I was trying to remember how long I've known Jerome, and I know that it's at least 20 years because here's us. Many years ago, 20 years ago, in fact, when we were working on our first project together, building a wood density database. And so at least 20, if not 25 years that we've been coming up with projects that we can do together. So it's an absolute pleasure to be here with a friend, a colleague, somebody who inspires me. I also want to thank both Ornella and Beatrice for taking good care of me the past few days. So this is my very first trip to French Guiana. And with these good friends, they took me around and made me feel welcome. And so I got to see many sites with them and with Jerome. So thank you. All right, so just a brief introduction to me and my group. I was trained as a botanist. I've long thought about plant functional ecology as well as plant macroevolution. I've built big databases in part with Jerome to look at some of these questions. More recently, I've started looking at termite and fungal functional ecology. Then this has been the big database work that a lot of my work has been surrounding. But then more the field part, what a lot of us do in the lab and where we go out to the field is thinking about how these organisms interact in nature. And so thinking about the plant life, but also the plant afterlife, how fungi and termites break down wood and how that's based on different wood compositions. And so I'll talk a little bit about that today with the experiment that I'm going to show you. And so for today's talk, I'm going to give you the background, a little bit of a 101 in wood decomposition. Very exciting. And then I'm going to take you through the history of the story. How did this story come to be that I'm going to talk on? The story itself. This has been a very unusual project for me, building a series of collaborators. And so I'm going to tell you a bit about the authors and then ending up telling you about the illustrator with these amazing pictures you'll see today. So I've been fascinated by why trees grow, where they grow in different parts of the landscape. And so to do that, a lot of that has been to look at how the wood is constructed. So looking inside the trees, the chemistry, the anatomy, the morphology of the trees themselves, and telling me why they can grow in wet versus dry places based on, say, the vessels. But more recently, I've started thinking about how does that construction translate to how they decompose. So why do some trees remain after a fire on the landscape, whereas others burn up? And so that's led me to thinking a lot about the afterlife of plants. Why does this matter? Well, plants are the largest above ground terrestrial biotic store of carbon. So there's vast amounts of carbon in forests. I don't need to tell you as an audience, since many of you are looking at this question. This is true here in my field site in Cape Tribulation in Australia. This is Mount Saro. You can see a beautiful tropical rainforest, but it's also true once trees die. And so wood can store that carbon and the nutrients in it for years to decades to centuries to millennia. And so if any of you have inherited your grandmother's favorite chair, you know that would last for a long time. What matters is if it starts to decay. And so the rates and forms by which carbon is released from wood are dependent upon who is actually doing that decay. So let me introduce you to some of these players. So globally, microbes, fungi, but also bacteria are important decayers. You find these everywhere you go. In certain places, insects become important. And so termites, especially in the tropics. where you get enough buildup of vegetation to carry fires. Fire can be very important. And then you can have other things like freeze thaw or solar radiation that can break down plant material. And so depending on where you are in the world, different players become important. And what determines, and that's because of the different environmental gradients. So the environment itself can affect who decays the wood and how fast. And I'm going to talk today about moisture and temperature. One of the things I'm going to introduce now is Q10s. So a Q10 is the rate of a process. So for a 10-degree increase in temperature, how much faster does a process occur? And this is built on the stoichiometry of enzymes. So typically an enzyme has it doubles in its rate for a 10-degree increase in temperature. So a Q10 of 2, a doubling of rate. Okay, and but what also matters is how that wood itself is constructed. So those traits that I talked about before. I'm not going to focus on that much today, but we think about it a lot in our lab group. Okay, and so here's a bit of the 101, the intro class. Okay, sounds good. So, I'll discuss the 101 of decomposition, and I'm going to especially focus on the biotic decayers. So those are the microbes as well as the termites. And so the first thing is they're very interesting because they're different in how they find the wood. So microbes disperse by spores and as well as colonizing with hyphae from the soil, and then they're latently present in wood as it's living. And so you can't predict very well whether any particular species of microbe is going to be in the wood, but you know that there will be microbes there. Termites, on the other hand, are searching animals. A worker has to go out and find the bit of wood to decompose. It has to go back to the mound and recruit its friends, its mound mates, and then it decomposes the wood. So it's a two-step process, a discovery followed by a decay. They're also different in how they release carbon in the wood. So microbes, as you can expect, don't have stomachs like us. So they excrete enzymes into the environment. There are cellulases and hemicellulases and things like that, as well as lignases that help to break down the wood. And those enzymes are dependent upon moisture delivering those enzymes to the substrate. Whereas termites do have stomachs, some of them have their own endogenous cellulases, others foster a suite of microbes in their guts, various archaea, that help them to break down the wood. And then others are, especially in Africa and Asia, are fungus farmers. They have a garden of fungus, a white rot fungus that helps them to break down the wood. Okay, now they're different in terms of their sensitivities to environmental gradients. What we know that in regional to local studies, typically microbes have a Q10 of 2. So it's likely that enzymes are driving the rates of decomposition by microbes. We don't know the Q10 of termites, but we know that they're largely tropical, as all of you can probably attest to. I've talked to a number of you today about termites that you find in your environment around here. And so we think that they probably are sensitive to temperature. I've already described how the enzymes need to be delivered to the substrate, and so it needs water. So we think that microbes are moisture sensitive, whereas we know that termites build mounds. They do sheeting where they cover where they are in the landscape. So they're likely, termite decomposition is probably less termite sensitive or moisture sensitive. Okay, and then finally, they're different in how they release carbon back to the environment. And so everything releases CO2 as it respires, but we know that termites are like little cows. They release methane back to the environment, probably by farting rather than belching, but they are releasing methane back to the environment. There's variation depending upon the type of food they feed upon. But everything that we've measured for termites so far has some methane signal. And methane is a 30-fold worse greenhouse gas than CO2, so it may well have important implications. Now, we also talked about how most termites can't break down lignin. Only some fungi can break down lignin. And if it doesn't get broken down, it's buried in the soil. So where the carbon goes in the environment matters based upon who's doing that decomposition. And that's really what we're interested in. What is the fate of that carbon? Is it contributing to greater warming? Is it getting buried in the soil? Okay, so that's my basic 101 of decomposition. So let's move into the history of the study. As I described to you before, I'm a botanist. I started my world by working in Ugandan forests, identifying trees and trying to understand where they grew, why they grew where they did. But I started building these trait databases. And so that led me to start collaborating with Hans Cornelissen and Will Cornwell in what became a working group that led to this paper, The Plant Traits and Wood Fates Across the Globe. So we reviewed how plant traits affect decomposition. I also started, and I was at the time in St. Louis, Missouri, so I started an experiment, a common garden, where I was looking at how different species of wood decayed in a place that was largely driven by microbes. I did find some termites there, but not very many. From there, we got some funding to start working in Australia with Jeff Powell and Will Cornwell in Sydney, where termites and fire start becoming important decomposers in the system. And then most recently, I had a project co-funded with Paul Eggleton and Steve Allison in far north Queensland. And with that work, we were looking at the relative roles of wood-feeding termites versus wood-dwelling microbes in terms of their effects on the decomposition process. Okay, and so we thought that the termites would be relatively more important in dry sites and in dry seasons. And so we asked, what are the carbon fates? What are the carbon rates? How fast is carbon turning over? What forms is it going up in the environment? And then we had some predictions of the future of far north Queensland, Australia in terms of the climate change, both temperature and moisture. So we're in the process of modeling, trying to predict the effects of incorporating microbes and termites into the future carbon cycling of the system. Okay, and this is we're working up at the tip of far north Queensland. We have a nice gradient with savannas, 2 savanna sites, two rainforest sites and a site in the middle. So a nice rainfall gradient that's about a 10-fold range in rainfall. And so as we were building this project up, I was trying to get funding and to NSF and to DOE, and I was getting responses back saying from NSF, well, termites don't really matter. They're not that big in the landscape, so why is this an interesting study for us to fund? And so what we did was with Lucas Cernusak and Alex Cheeseman, we ran a pilot study at our endpoints where we wrapped radiata pine blocks, so it's a novel substrate for all of the termites and microbes in Australia, wrapped it in mesh and then cut holes in the bottom. And we put it out in the landscape. And what we found were differences through space and time in terms of what moisture did in terms of the rates of decomposition. So this gave us a pilot study that then we turned into a big NSF NERC grant that just wrapped up this past June, although we're in the process of writing papers. So we did things like measure the amount of biomass in the landscape at all of these sites, both the living and the dead wood. We knew that there were these amazing termites in the system, Coptotermes acinasiformes. These hollow the insides of the trees. If anybody's ever heard a didgeridoo played, these are termite-made musical instruments made by indigenous people in Australia. So it's the termites that are hollowing the inside, and we're harnessing that for music. And so we wanted to understand how much biomass was missing before the tree even senesced. So we've done that in a number of studies by using this resistograph drill to drill holes into the trees. We've cut down trees. So my PhD student, Abby Yatsko, is working north of our field sites with people that do TLS, terrestrial lidar scanning, so we can look at the amount of biomass in the forest, and they've cut down trees to look at the inside, how much is missing. We've put out woodblock experiments. Here's my former student, Becca Clement, collecting termites along the gradient so we knew who's there. We've measured CO2 and methane with infrared gas analyzers in the wood blocks itself, as well as in mesocosms where we put termite mounds in buckets. And also, this is part of Abby's work where she's going out to termite mounds and putting collars on and measuring them directly in the field. So we're learning a lot about the system and how carbon cycles and when it's through termite process in a microbe. But we weren't satisfied to just stop there. That tells me something about Queensland. But I, of course, wanted to go global and think what happens around the world in terms of the contributions. And so when I was setting up the project, I happened to be in Australia and chatting with somebody who was head of TERN. So these are the super sites where there are flex towers and a whole bunch of vegetation measures, but they weren't measuring the loss of carbon in the system. And so the Turn Super Site in Australia put out our woodblock experiment, our pilot study across there. I was in Bordeaux on sabbatical at the time, and I was giving a lot of talks sort of like this, and traveling around. And as I talked to people, they were really interested in thinking about trying to link together different sites around the tropics and then eventually around the globe to think about the different contributions of microbes to termites and decomposition. And so I got a lot of sites. I looked at the map and I said, okay, we have some holes. So then I went on social media, Twitter and Facebook, and got even more people to join in. And so we ended up with a repository that was shared amongst all of us, and recruited people to look at this question. You can see here, so we ended up with 108 collaborators at 133 sites in 20 countries and 6 continents. If you go on Twitter, here's Alberto with his dad hauling wood up into the Alps. Here's another PhD student in New Zealand. So we had lots of blocks going out into the environment. Okay, so that's the background of how we got to this story. Now let me tell you the story. So this came out last year. We made the cover of Science, which was quite fun, with termites. And so these are Microceratermes from the rainforest in Australia. You can see the amazing list of 108 co-authors that were part of the project. Even more fun than making the cover, I think Beatrice sent this out to the PhD students. The First Dog in the Moon, which is a political satirist for The Guardian in Australia, wrote up our study. And so the question was, termite scientists, is there a better job in the world? No, and I'd agree. This has been quite an amazing journey to learn more and more about termites. There's one thing, they got the rainfall pattern wrong, but otherwise it's very accurate to our study. So have a look at the cartoon if you want. Okay, so what we were trying to look at was the relative climate sensitivities to temperature and precipitation of these processes. Our expectation was that microbial decay would be highest in warm, wet locations for the reasons I described, both the Q10 of 2 that we expected, as well as the enzymes needing to be delivered to the substrate. For termite discovery and decay, we thought that it would be highest in warm locations because termites are largely in the tropics, and we thought that they would be less sensitive to precipitation. So here you can see the distribution of our sites. We have certainly big gaps in the map, but we have a really good distribution latitudinally. We have equal representation in the northern and southern hemisphere, equal representation in the tropics and the temperate zone. And what we did was we used the radiata pine again, wrapped in mesh that excluded termites, and then we cut holes in the bottom to allow termites in. And each person put out 20 stations with replication of one that allowed or excluded termites for each pull. Everybody did 1 year. Some people did 6 months, many people did 2 years, and some places did up to 4 years. So we had a range of how long this ran. And I should say that Jacques Couchon has deployed this here in French Guiana. So we actually represent our study, or the study here is represented in our global study. You can see that here's mean annual temperature on the x-axis and mean annual precipitation on the y-axis. And you can see that we did pretty good coverage around the globe. We had nothing in the tundra, not so much in the boreal forest or the temperate grasslands and deserts, but otherwise pretty good coverage in the different climate zones. Here you can see these are blocks. At the top are 2 blocks from Australia. On the left is ones that went through termite decomposition, and the right was microbial. And I should say that we didn't exclude microbes, so the microbes were always in the system, and then in some cases we added in the termites. The bottom is from the pilot study. This was the block I put out. It weighed 180 grams. When I harvested it, it weighed 300 grams, and only 3 grams of wood was left. So in 1 year, there was a massive amounts of decomposition by termites. Okay, and so to answer our questions, for decay, we looked at the difference in the final mass by the initial mass over time. We separated out those blocks that were discovered by termites versus those that were undiscovered. So undiscovered, just a microbial pathway, discovered, termites plus microbes. And then for discovery, we had 20 stations that were allowing termites in, so termite included, and we counted how many blocks that termites actually found of those. We used mean annual temperature, mean annual precipitation, and its interaction in both models for decay and discovery. Okay, so that's the setup, pretty simple, although analyzing all of those blocks and bringing in everybody's data was quite considerable work. And so what we found, we're going to start with microbial decay. On the x-axis is mean annual temperature. On the y-axis is the decay constant K. And what we saw was that microbial decomposition was only sensitive to temperature, not precipitation or its interaction, which was really surprising. And I'd be happy to talk to you a little bit about that. You can also see that there's a positive relationship, that as temperature increases, you get faster decay by microbes. The Q10 was 1.7, but not significantly different to 2. So we essentially recovered what regional and local studies have found was this doubling of decay with an increase in temperature of 10 degrees. If we move to termites, again, mean annual temperature on the x-axis and decay constant on the y-axis, what you can see is that termite decay was not sensitive to precipitation or its interaction. It was only sensitive to temperature. The other thing you'll notice is that it has a Q10 of 6.9. This is huge. We reran the analyses, not believing that termites were this sensitive to temperature. So they're about fourfold, termite decay is about fourfold more sensitive to temperature than microbial decomposition. It's really surprising. And then finally, probably most interestingly, here's discovery. So you have, again, mean annual temperature on the x-axis, y-axis discovery. And what you can see is that the discovery is sensitive to both temperature, precipitation, and their interaction. And so with increasing temperature, you have higher discovery. What you see is that the wet sites are in blue and that there's a shallow positive slope where you have higher discovery in wet places with higher temperature. But what was really interesting is the golden color. These are our dry sites. And you see this really, really low discovery at dry, cold places, but a really rapid increase in discovery as you cross into warmer and warmer places for the dry sites. So the highest overall discovery was in the hot, dry places of the world. And I didn't tell you what the colors meant before on these dots. What you can see is that low discovery is yellow, and the red to almost black are the high discovery. These arrows here denote the tropics. So you can see that there's more red and black as we shift into the tropics. You can also see, so the green in the top is tropical rainforests. We have subtropical seasonal forests and savannas. And then we have subtropical deserts. And you can see that the darkest colors are in these drier sites, so the subtropical deserts and the tropical seasonal forests and savannas. If we put the two pieces together, the discovery with the decay, so here we have the different biomes from boreal zone up to the tropics and the decay constants. The box on the left, the boxes, the solid lines are where it's just microbial pathway. The ones with the dashes are where you have termites plus microbes. And what you can see is that termites, when you add in termites, anywhere they're found, they always elevate the rates of decomposition. Where they elevate the most are these drier sites, so the subtropical deserts and the tropical seasonal forests and savannas. So interesting, we've said something about how microbial decomposition and termite decomposition is affected by termites or by temperature and moisture around the world. But we didn't want to stop there. We wanted to think about the future world. And so we're saying something about the sensitivity of termite discovery to temperature and moisture. And so what we did was we wanted to think about what happens in the future. So we took our discovery model. We said that you have high discovery if there's 50% or more of the blocks are discovered. And then we combined this with a WorldClim downscaling of CMI P6 climate models. We had a stronger model where things are going to get much worse by end of the century. So an SSP 5-8.5, and then a weaker model, SSP 1-2.6, where things are getting possibly a little bit better. And what we were predicting was what would happen by mid-century. What you can see is in the top panel is a stronger model and the bottom panel is a weaker model. In gray is where there's currently almost no termite discovery, and that's going to continue in the future. In the bright green, termite discovery is low now and it's staying low. In the olive green, it's already high, it's already above 50% and it's going to stay that way. In buff, it's outside of the constraints of what we've measured empirically, so we're not predicting. And then the interesting one is the one in red, the orange box. So this is where by mid-century, currently there's low discovery, less than 50%, and we're expecting discovery greater than 50% by mid-century. And so you can see In the Southern Hemisphere, especially in Africa and the New World, we're expecting higher discovery by termites. This is also true where I live in Florida, we're expecting higher discovery. So we expect the footprint of termites is likely to increase under future worlds. And you can see this. So here we have the increase in terrestrial area with termite discovery that's high, and the global mean terrestrial warming. So from 1 degree up to 5 degrees. And you can see a strong positive relationship under both models. Okay, so what we were able to show is that microbial decomposition was highest in warm places, but not wet places. And that termite discovery and decay was also highest in warm places, but interestingly in dry places. These hot, dry places, and a large predicted increase or expansion of high termite discovery by mid-century. Now, this is only based on temperature and moisture. There's a lot of other things going on. Obviously, land use change and things like that will affect where we actually find termites and their ability to move. Okay. So that's the story. It was an amazing collaborative effort. So I'm going to tell you a little bit about the authors. And so I think one of the things that's really important to me for my science is to make it collaborative, inclusive, and open. And so we came up with a very simple protocol. We had no organized funding, no funding across the entire project. Each PI or each person came up with their own funds. I had a PhD student in Brazil, and then I had collaborators from the US and Spain with 20 sites that they were able to put in. So people could join in at different scales based upon their budget and their abilities. And so that let it be accessible to many. It was bottom up, as I said, no group funding. Everybody had their own small resources that we put together. I recruited by word of mouth initially and then by social media. So I had encounters with people. Much of it occurred across COVID. So I was sitting at my house communicating with people around the world as they joined into the project. I offered co-authorship to everybody who was involved in the project, wanting to make, this took a village, and I wanted to recognize the village. So we had 108 co-authors. I spent the day before when we went live trying to look up as many, everybody's Twitter handle if they had one. And so I tried to call out everybody who helped to contribute to the project. It was really amazing. So it was across COVID. I never met most people, never mind one another. And so I had 2 group Zooms where we met 12 hours apart. And we had 6 continents all represented at one time on our Zoom call, where people were able to share their experiences of what they found with the wood blocks, the animals that were eating the wood blocks and things like that through time. So pretty amazing. And I've never been involved with anything quite like that before. Let's see. Yep. Speak up. Yep, sorry. I'll talk a little bit louder. So the other thing is that it's really important to have open science so that people can repeat what you've done to check how you've run your analysis, but also to be able to come up with new creative ways of using your data. So the data are all up in Zenodo and Figshare, so you can download them and play with them and try them for your own purposes. And so finally, I'm going to talk about the illustrator. So I've shown you a series of amazing images. These are by Donna Davis, who is an artist who worked with our project. You can, I put that twice, so you can see, you can go to her webpage. I found her because I was in Australia at the time setting up the initial project. And she she had a display on where she was looking at mycorrhizal interactions with plants in Australia in an endangered Melaleuca forest. And so I heard words like mycorrhizae and fungi and trees. These are spore prints that she screened onto silk. She had various interactive exhibits that you could see. And so I was really impressed by how she could reach a different audience with her message than I could reach with mine. And so we recruited her on. We've done a series of, she's displayed in a number of art galleries. She came out to the field with us before COVID and then again in 2022, last year. This is Steve Allison, one of my collaborators, with a silk print of a bunch of termites printed on that he has wrapped around his shoulders. So her images are really stunning and evocative. She hasn't, because we were doing this on COVID and internationally, and we wanted to be able to all display her images. A lot of it is digital. So you can see different videos of swirling termite mandibles and fungi and plants. You can also watch, this is showing decomposition of these plants next to an old machine that measures the humidity of the system. As I said, she's displayed at a number of galleries. We've participated in a number of science artist collaborative discussion panels, and it's really interesting thinking about how similar our processes are for creative discovery. So it's really been a joy to think about our system with her and with others. And let's see, is this? Yeah, so this, we've also had a digital exhibit. So if you want to go to the University of Miami has an art museum where we have this exhibit up. This is one of my favorite pieces where you have swirling termites, plants, and microbes. This is actually a graph of termites. So it's pictures that my student Becca took of termites. And you can see that they're more concentrated in the bottom. So it's showing that at the driest site, you have more termites than the wettest sites. And this is called mirror, well, originally called mirror, mirror on the wall. So thinking about what do we call fair and beautiful and often gilded and bright and shiny things. And so these in this case are termites. Okay, and I think it's really important to learn about being able to communicate your science to others. My students have all worked with Donna across COVID. They've all become incredible at being able to visually and verbally display or describe their science. So we have various podcasts and videos up. And here's one where Donna worked with a digital artist to try to portray what we think termites might sound like or microbes might sound like as they're decomposing wood. Okay, so that's the story. That's the co-authors. That's the illustrator. I wanted to make a plug because here's Baptiste, a new postdoc in the lab, and we're trying to look at what's on the inside. So what happens to plant material once it passes through termites? We're currently collecting the inside of termite mounds. You need to hopefully be able to identify the termite that you're working with. So far, he's gotten from 20 sites in 6 continents. So if you're interested in joining us, let me know. And with that, I'll stop and take questions. These are a series of fun facts that Becca wrote in collaboration with Donna, who made the images. And we're in the process of recruiting. I'm increasingly working in Brazil and Chile. So if you know anybody looking for a PhD scholarship, let me know. So I'll stop there and take any questions.
Thank you, Amy. Any question from the audience? Yes. Thank you very much for the very inspiring presentation and great results. I was wondering what is known about the ratio between methane and CO2 produced by the composition of wood by termites.
Right, it's a really good question. So when we think about methane emissions from cows, the methane is being released directly into the atmosphere. Most termites are living in mounds, and so as they're releasing methane, what a lot of people have found is that the termites may release methane directly, but then the mounds themselves have methanotrophs that scrub the methane and convert it into CO2, in that conversion process. So just because termites are producing methane doesn't mean it's being released to the atmosphere as CO2. And so you can see, you saw that termite mound with a whole bunch of circles on it. That's where Abby's trying to look at directly the fluxes out of the mounds of both methane and CO2. And so a lot of the studies that have measured termite methane and CO2 emissions are doing this on individual termites. There's even a global database of that. But what we're trying to do is think about how the soil or the worked material that termites build around them actually hosts these methanotrophs and what the actual flux rates are out. So this is Abby's dissertation work, and others are working on this too. So I can answer that question better probably in about a year. So I'm happy to come back and tell you next year. Thanks for the great question.
Thank you. Anyone else? Questions? Over there.
Yes, thanks for this very interesting talk. The question is, don't you think there will be a threshold, a maximum level of temperature where the Q10 will become below 1?
Yeah.
Because it will be too hot for them?
Yes.
And
Yeah.
I've got another question that's more complicated. And so
You can ask that if you want to. So let me start with the first question. And so, yes, it's interesting. So I didn't actually tell you all that's under the hood with that. So our pilot study that we ran, we did that in, I think it was a La Nina year for Australia. So it was really, really dry. We had a failed rainy season. So the savanna sites got no rain. We had no discovery by termites across the dry season for both rainforest and savanna. It wasn't until the next year's rains kicked in that we started getting some termite discovery. And then we found that termites were in our Australian site equally sensitive to, they increase the amount of decay both in our rainforest and savanna sites equally. So I think there's a lot more nuance. It's not just that termites are better than microbes. They are also very sensitive to temperature. So you're exactly right. We've actually measured some of that. It's you can look under the hood in all of these different relationships, a global pattern, but what we've measured in Australia is exactly that. It's a great question. Thank you for pointing that out. I don't know if you have a Jerome.
I have a question. I'm sure you answered this question a million times already, but if you were to pick another species than radiata pine, like oak or like a tropical species, like, this wonderful wood samples from the tropics, what are you expecting to find?
Mm-hmm. Really good question. And actually, Jacques did that. He put out radiata pine, but he had a number of native species here too. And I can't remember we can ask him what he found with those. In our Australia project, we've put out radiata pine, but we've also put out native species. In our Australia work, what's interesting is that the termites in the savannas like radiata pine more than they like the savanna species. So that's some of the the eucalypts and Combretaceae and things like that. And the rainforest, they like the pine less. So this is where our knowledge about plant traits and plant construction really matters. And so I think that you'll see differences in what the microbial and the termite preferences are. And so we're using this as a proxy to start to estimate how global decay happens. But I think you really need to know those interactions with the plant traits and that construction to see the preferences of the termites and the microbes. You can come up with, and Sebastian Sebold has looked at insect decomposition overall, and he's put out different species of conifers and angiosperms to look at that variability, but it's really critical to look at that. Thanks, Jerome.
Thank you. Any more questions in the audience? Don't be shy. There is no bad question. Yeah, go ahead.
I'm going to have to leave it, I think, around 2 p.m. today, so please ask now or catch me at lunch. So
okay, so you have tried to project the discovery of termites and predict how global change will affect this. But what about wood decay itself? Do you predict higher rates of wood decay? And if yes, what's consequences on ecosystems?
Right. So actually, this is what Jerome and I spent a bunch of time in our drive back from Kauru yesterday talking about, is thinking about the rates of decay and how they might respond. And so because of that Q10 of 2 for microbial and that Q10 of 6.9 with termites, I expect that you're going to get faster rates of decay with warming. Moisture will do different things. There's some things that are probably under the hood that mean annual precipitation is probably not a great way to estimate how much moisture is in wood. Wood has bark and things like that can lock in moisture. It can take a while to invade. So I think that there's the soil moisture, or even better, wood moisture is probably a better precipitation estimation. So I think combining both the temperature and the precipitation, and I think moisture will have an effect. It just didn't come out in the wash in our study. So I expect it to increase. We were talking about what are the rates at which tree fall is happening. And we've shown that as you're a snag and you fall on the ground, we expect those rates, at least in the US, to increase. And so if bigger things are falling, it might take longer for them to decompose because log size matters, but that you also, generally we're expecting faster decay rates through time. Really good question.
Any more? Great. I'd like to run, but I don't want to fall.
Giving Jerome his exercise.
Hello, thank you for your interesting presentation. My question is a little bit based on this fun fact on the right, on the lower right, about the interaction between ants and termites. So my question was, how will this develop with climate change and with the increase in termites due to all these factors?
Right. So, so the biggest predators of termites are ants, and termites have amazing adaptations to repel ants. Some of them have like glue, or they paint acid onto their ant predators. Some of them have, my collaborator Paul Eggleton has said, like Swiss Army knives where they lop off the heads of ants and things like that. So there's all sorts of ways that they protect themselves, and that variability might differentially affect them in terms of how they respond to ants. Some of them withdraw into the nest, which is also, we've often found that when it's really hot, like in Australia, it can get to 40 Celsius. We don't find any of the termites in the top of the mounds. How that interaction happens, Kate Parr and folks in her group have done some amazing work looking at the thermotolerance of ants, and others have done this as well, knowing that ants also are responding to the temperature and are sensitive to temperature differentially. And I think that some of the work has shown that there's more variability or more robustness in changes to temperature in towards the poles than at the equator, where there's less variation in temperature through time. So I think it probably matters where you are in the globe. And then we have maybe novel ants and termites meeting, how they interact will be interesting to see. So that's a very evasive answer, but I think it's interesting to think about how those interactions, those biotic interactions will change under climate change.
Great, thank you.
Thanks, sir. Is there somebody back
Any
there?
more?
No.
Any more questions? Well, if not, thank you very much, Amy, for this wonderful presentation. And it's just about time to have a lunch break. But before this, a big round of applause to Amy again. Thank you.
Thank you.